my name is Fatou Mbaye, and here is my protein project for Unit 2. Here I was assigned the melanocortin 1 receptor, or otherwise known as the MC1R protein. Its mutation focuses on amino acid number 67, otherwise known as arginine. Later on, the mutation will replace arginine with cysteine. The MC1R functions as a receptor protein. It is a transmembrane protein receptor that becomes stimulated after contact with hormones. This is a receptor protein due to being located on the surface of the plasma membrane. In this case, it is the melanocytes. Additionally, receptor proteins signal cellular pathways. In this case, it is the signaling of eumelanin and pheomelanin. The normal function of this protein is to produce pigments of eumelanin and pheomelanin in order to give an organism its pigment. This protein is normally found on the plasma membrane. So here I have the protein, and then I have the semi-permeable layer. Above is supposed to represent the outside of the cell, and below is supposed to represent the inside of the cell. The type of cell that this is found on is the melanocyte cell. And additionally, it is generally found on animal cells. So in this case, the protein became less active. So this is how the mutation has an effect on the protein function. The original function of the protein is to produce eumelanin with the occasional addition of pheomelanin. When the mutation occurs, the MC1R protein is unable to bind extracellular melanocyte-stimulating hormones, otherwise known as AMSH. This results in a blockage and a lack of eumelanin production. Furthermore, the phenotype of the organism results in a lighter than usual pigment. So, I will now show you the mutated protein. So, here you can see a change. Instead of the orange puffball, which represents arginine, I have replaced it with a purple puffball, which represents the protein change of amino acid number 67, and it's now become cysteine. So how does this change the structure? How does the structure impact the protein's function? So in the original protein, the amino acid contains amino and carboxyl groups in the main chain and a guanidino group in the side chain. The properties of the original amino acid side chain are positively charged, polar, hydrophilic, and compared to other amino acids is quite large. The bonds that the original protein, the amino acid of the protein can participate in are hydrogen bonds. The properties of the new mutated amino acid in terms of functional groups are carboxyl groups, amino groups, and sulfhydryl groups. In comparison to the mutated and original, the mutated version contains a sulfhydryl, while the original does not. The differences in the type of bonds that the mutated amino acid side chain can participate in is that the mutated amino acid can participate in disulfide bonds, while the original forms hydrogen bonds. In conclusion, the mutation affects the protein function, the function of the cell, and the function of the organism by amino acid number 67 being altered in terms of the sequence of protein, and this makes the protein non-functional. It affects the cell because the mutation forces the cell to create no or less eumelanin. And finally, the function of the organism in turn results in a lighter than usual pigment, which is expressed in the organism's phenotype. Finally, the limitations of my model include the fact that I cannot include each of the 317 amino acids of the protein. Additionally, I cannot include the details of each functional group. Thank you.